Hi, I'd like to welcome everybody to the Leaky Gut publication discussion panel here today. We're very excited to be discussing the upcoming publication associated with these data sets from the study that was run here. With that, I'd like to hand it over and go around to our panel to introduce themselves accordingly. Jason, it'd be great if we could start with you.
Great, thanks, Will. So Jason Bush, I'm a scientist with MSP Starch Products. I've been responsible for the R&D associated with Solnul, the prebiotic ingredient that was the subject of the study.
Wonderful. And then with that, we'll move to Dave.
Hi, I'm Dave Schibli. I'm the Associate Director of the University of Victoria Genome BC Proteomics Centre. We are an analytical facility performing mass spec analysis in the areas of proteomics and metabolomics. And we've been working with Jason and Solnul for almost 5 years, performing metabolomics analyses, including for the study we're talking about today.
Fantastic. And Josh.
Josh Baisley, Senior Vice President of Clinical Operations here at Nutrasource. I work closely with Jason on the study design, the regulatory strategy, and then oversaw the team that executed the trial. Wonderful.
Okay, then with that background, we'll move into some of our key discussion points, and we'll start from the foundational piece. Jason, from your and Solnul's perspective, what was the background that really led to this type of research? What was the objective, and what brought you as a firm and as the chief science officer to say this is where we want to go?
Yeah, so Solnul is a resistant starch, and resistant starch is a type of ingredient has been known for quite some time, including its role as a prebiotic. The dose of resistant starch that's previously been studied was always really high, like 30 or 40 grams. And so we started kicking around the idea that if resistant starch was really such an important prebiotic, then it's likely going to have effects at doses that are comparable to other prebiotic ingredients. And so that's where the discussion kicked off with your team and with Josh. You know, how can we design a prebiotic study to substantiate that claim for Solnul as an ingredient? And, you know, as you know, a clinical trial is quite the investment. And thinking about what information you want to capture can really dictate the expense and kind of the work that goes into that. And so we were at that time, you know, obviously we're interested in changes in the microbiome, changes in stool form. You know, those are important details to have for prebiotic claims. But around this time, probably talking 4 or 5 years ago now, people were starting to think about the gut-brain axis, the gut-skin axis, and so forth. And with a prebiotic, right, that's something you ingest, it goes into your microbiome. And in order for other parts of the body, you know, the brain or the skin to actually achieve a benefit, the activity of the microbiome needs to leave the gut, right? It needs to transcend that gut lining. And so we thought, well, if Solnul is really this great prebiotic, that it's as great as we think it is, it should be having effects beyond the gut. And so we kind of doubled down. We said, well, we're going to pull blood samples from these participants at these crucial time points, store them on ice, and then, you know, work towards analyzing them. And that's where conversation with Dave really unfolded. Is he was really helpful in crafting the grant proposal that helped us secure funding from the Canadian Agricultural Partnership, Ag Action Program. And that was really what unlocked the opportunity for us to take those samples out of the freezer and then do metabolomics research on them.
Great. And to that end then, Dave, switching over to you, it'd be great to get some insights as to what exactly metabolomics research is and why there's so much excitement and buzz in this ingredient category and in other ingredient categories related to this area of science and research.
Yeah, so I think many people are aware of what genomics is. They've heard about it in the news. You know, genomics is the study of the genes or the genome of an entire organism's DNA. Metabolomics is the functional step beyond genomics. It's the large-scale study of hundreds to thousands of small molecules, commonly known as metabolites, within cells or fluids such as serum, tissues, or organisms such as bacteria. Individual metabolites can include common compounds that people normally think of, such as sugar, like glucose or vitamins. However, when we're talking about metabolomics, we are going beyond the simple measurement of an individual or a few compounds, and instead measuring hundreds of interrelated compounds simultaneously. So to put back in context of genomics, you can think of an analogy of cars on a highway. City planners could design a series of highways, roads, on and off ramps, and this plan can be considered an individual's genome, a picture of what a system is and how it should work. But this is a static picture. What is missing is the functional implications of what happens on those roads when there are cars, buses, pedestrians, and furthermore, what happens when there are perturbations in the or errors in that plan. An accident occurs, an off-ramp is shut down, a speed limit is changed, or a new type of vehicle is added. To understand that, you look at all the different traffic on the road. So metabolomics is the equivalent of that. It is the actual function of the system based on the original genetic plan, plus all of the additional inputs and outputs, such as from food and environment. For example, returning to glucose metabolism, we can measure how glucose is used in glycolysis and track its metabolism through more than 100 different interrelated metabolites and pathways, and eventually which provides energy for your body from the food that you eat. So we've been doing this work here at the Proteomics Centre for more than 40 years. I think everyone understands that genomics is having this huge impact on personalized medicine or precision medicine. However, genomics is only telling us part of the story. In many cases, like I said, it's a static picture of what's going on. It's not actually telling you what's happening. So we perform metabolomics analyses to understand the complexity found in natural pathways and systems in order to ask questions how metabolites are synthesized or used by an organism. Metabolomics is providing us information on the phenotype of an organism under different conditions. By looking at entire pathways, we can get an amazing amount of knowledge and insight to what is actually happening in individual systems. So we can use this information to understand changes related to disease or disorders, which can then be used to identify treatments that will bring those pathways back to a normal, healthy state. You can also understand the reverse of that, using metabolomics to understand how a treatment is working and what other impacts it may be having and how it can be further improved. So metabolomic pathways are highly complex, interrelated. Taking snapshots of individual compounds in isolation can give a misinterpretation of the results or not provide enough information to properly come to conclusions. So like what we've done with Jason and Solnul in this study, where multiple different factors could lead to similar results, it's essential that we dive really deep into the biology to interpret possible mechanisms, actions, and come to viable conclusions.
Great. And are you seeing an uptick of activity at your center beyond some of the normal channels of activity into the nutritional space recently? And what does that look like for you guys?
So, like I said, we've been doing this for a long time. Human health research is probably the 90% of what we do here at the center. Historically, that's include basic research into the biology of disease, a lot of studies in traditional pharmaceutical drugs and things like that. But we are definitely seeing more and more interest in things in the nutraceutical space or similar type products. But it's still definitely a small part of it. And this study that we did with Jason is probably not only the biggest nutraceutical project we've ever done, it's probably one of our biggest projects we've actually ever done, just in the breadth of metabolites that we've looked at. What we're seeing is we are seeing some interesting companies come in similar space that are looking not just at disease interactions of metabolites and how their therapeutic is working, but going beyond that, also just the manufacturing, quality control, regulatory types of things. So people are looking at compounds similar to this or products similar to this. Is what they're presenting to the regulators or customers actually what they think it is? What's the composition of it? What's the quality over the long term? And we're finding that this is actually a really essential thing for complex products when you're getting into that regulatory piece and going to the FDA for approval or similar regulatory agencies. That what you're selling is actually what you say it is over time.
No, makes sense. And then specific to the study, Jason, back to you. Can you kind of give us an overview in terms of the output and the data sets of the key findings of this publication?
Yeah, you bet. So we've kind of rolled out this information in stages. Josh and I worked on the first publication, which really described the prebiotic benefits of Solnul coming from this study, really around increases in bifidobacterium, Akkermansia, as well as improvements in stool form. And we wanted to kind of build on that in this subsequent publication. And as Dave mentioned, we get a real bucket load of data when we look at metabolomics. And so as we were sifting through that data, trying to kind of figure out what the most exciting aspects of it were, we really landed on reductions in histamine. So that's kind of the one key takeaway from the study, is that people consuming 3.5 grams of Solnul saw a significant and meaningful reduction in the circulating levels of histamine. Now, just in case people aren't familiar, histamine is a marker of inflammation, and it's actually a mediator of inflammation, right? So when you have an allergic response, histamine is released by cells that cause more and more other immune cells to kind of come and release more and more, and it kind of amplifies the signal. So there's a lot of interest in reducing histamine from an allergic response, but also from a systemic inflammation response. And so we kind of picked that as our trailhead, and we kind of used that to open up investigation into metabolomics. And the really cool thing about having this data set is that it allows you, even though those participants donated their blood to the study years ago, we're able to ask new questions about it. So why would Solnul lead to a decrease in histamine levels? And so the first thing we chased down was, well, maybe it has to do with histamine intolerance. Maybe these individuals don't have enough activity of the enzymes that normally break down histamine, and when they take Solnul, it's helping them kind of break down histamine in that respect. Now, one of the cool things about the study is that we were able to disprove that story. So it has no effect on those enzymes. So that's one of the interesting things about doing science, is that you can have a great hypothesis and be wrong, and it can lead to other great hypotheses. So one thing I didn't appreciate going into the study is that most of the histamine in our body, it's not really playing a role in the immune response. It's actually trapped within our gut. The foods we consume, typically, fermented foods, meats, cheeses, things like that, tend to be high in histamine. And furthermore, friendly bugs, friendly bacteria like lactobacilli will actually produce histamine. And so this got me thinking, well, if it's normal and important for that histamine to stay within the gut, what if Solnul, which is being ingested as a prebiotic, is actually acting locally? What if it's helping to enhance the intestinal barrier function? We all hear about leaky gut, right? This idea that as we eat toxic foods or inflammatory foods, that the barrier between the cells of the intestines is kind of falling apart, and substances normally in the gut are leaking out. Well, histamine is a great example of a substance that can be a marker of leaky gut. And so we kind of methodically worked through the data by looking at previous publications on leaky gut. And we were able to find that acetylated polyamines and a certain group of amino acids that are typically elevated in leaky gut patients are actually decreased in people consuming Solnul. So that kind of helped solidify this hypothesis. And then maybe the most surprising finding from the study relates to short and medium chain carnitines. And I won't go into detail, but these are substances that build up when your mitochondria, those engines in all of our cells, aren't working properly. And one of the kind of best characterized aspects of leaky gut comes from people taking things like aspirin, ibuprofen, NSAIDs. And these cause leaky gut by impairing the mitochondria. And so when we look at our data, people consuming Solnul had significant reductions in these in these byproducts. And the interpretation there is that Solnul somehow, and we're still working this through, that is enhancing mitochondria function. And so when we look at the totality of the evidence, we see reductions in histamine, reductions in acetylated polyamines, amino acids, and improvements in mitochondrial function. The data that we've collected here, the metabolomics data, really presents a compelling case to support that Solnul is improving intestinal barrier function.
And with the leaky gut concept, because there is a lot out there on leaky gut, and thanks for that explanation specifically on what it actually means and why you might not want it. But how would you assist with improving it dietary wise, lifestyle wise? What are the symptoms for the individual that they can't say conclusively, but they may have an inkling that they're affected or impacted by leaky gut? What would show up in someone's daily life that would tip them off that this could be an issue?
No, it's an interesting question. And I was recently at a talk where somebody said, the vast majority of people in this room likely have some level of leaky gut. And so it's not an on-off switch, a diagnosis that's easily made. But I think the hallmarks of leaky gut tend to really circle in around inflammation. Not only that movement of histamine out of the gut, but also the body's reaction to having this either partially digested food or even waste products entering into the bloodstream. They're kind of responding to that through typically low levels of inflammation.
Got it. So that could be one factor or one signal that perhaps an individual has leaky gut is just level of inflammation, I'd say around bloating or other symptomologies.
Absolutely. There are more directed tests that can be conducted that look at levels of proteins, things called zonulin or occludin. These are proteins that typically call them cellular rivets that actually bolt those intestinal cells together to make sure that there's no leak in between them. Now, that was kind of outside of the scope of our investigation. You'd really need to do directed assays on that type of study. But as you and I have discussed, there's always a need for further research. Absolutely.
Okay. Now, Josh, in terms of running the study and helping with the design, etc., back to the consumer, in this case, the participant population type question, describe a bit the importance of the population that was selected, why that population was selected, and describe it as a cohort for this specific study.
So for this particular study, we were really focused on generally healthy adults. Again, this is who this product is going to be marketed towards, so we want to include them in the study design. But also from a regulatory perspective, we really can't go into the disease state. You can get into the slightly sick, but you don't want to be in the fully sick or diseased, just due to the regulatory burden that has, as well as can really change what the product is. And the intent here is that this product is a dietary supplement, so we want to play in that space. Now, there are a lot of confounders when it comes to looking at the metabolomics and these proteins. So part of the design too was looking at washing participants out as much as possible of other dietary supplements and vitamins and minerals, things that they don't necessarily need to be taking. They're taking these to support their general health, but they could go off of these for a period of time to participate in the study. So we actually had a 2-week washout period from all vitamins, minerals, and other supplements. Of course, food, we can't do anything about that unless we put them in a controlled diet for that whole period, but then that has cost implications as well. So that was kind of the structure of what we were looking at from the participant perspective. Just again, keeping it broad enough that it's targeting the right participants from a marketing perspective, but eliminating some of those other factors that we knew were going to bias or impact our results.
As a follow-on to that, what does this mean in terms of the results interpretation that Jason just shared based upon the fact that these were predominantly healthy individuals based upon the criteria?
From a regulatory perspective in the classification of this product as a dietary supplement, that obviously bodes very well for Solnul. Again, we're not looking at disease. We're looking at supporting healthy individuals and supporting the natural state of the body. So from that perspective and claim substantiation, that's again why we want to keep this in that healthy population. So seeing changes in that population is what is good from the claims perspective, but it's also very important because it is harder to see those changes when you are healthy. So getting these marked changes is a great thing for Solnul, and to use that both marketing and for claim substantiation.
Yeah, Will, if I could follow on that, I think that's one of the things that I think excited people about this publication is that we're able to see such meaningful improvements in a healthy population, right? We generally think that a healthy person isn't going to have high levels of histamine. And for us to have the reductions that we saw as a consequence of consuming only 3.5 grams of Solnul was actually quite surprising. And I think that from a future research perspective, that gets me really excited. If you do a study in type 2 diabetics, you got a lot of room to show improvements in blood glucose. That's a known detail, right? Yeah. But when we look in healthy people, I think what this research is starting to tell us is that there's a lot of room for improvement that we had no appreciation for. In other words, there's a lot of unturned stones for our industry to address, the keeping healthy people healthy concept, but still do meaningful research.
Absolutely. No, excellent insights. Thank you. And then, Dave, in terms of this type of work, you touched on this a bit earlier. This was one of the bigger ones that you guys as an institute have ever taken on. Are you seeing generally then a trend towards more activity in the nutritional space into the future from the standpoint of the increase you're getting and where you see some of this going?
Just so, in general, we're seeing a huge uptick in metabolomics projects. 4 or 5 years ago, this was a relatively minor space for us. Over the last 5 years, it's probably grown 5 to 10-fold.
Wow.
And, that is definitely the nutraceutical piece is growing in there as well. The other interesting thing, and just listening to Jason and Josh, what we're seeing is, on this gut impact on health. And that's a huge piece that's growing right now, more and more projects in that space and the impact of what bacteria can do on just the entire body. I would say we're getting, 3 or 4 projects a month right now, just in that one space alone. So I think there's some huge potential there on whether it's nutraceuticals or pharmaceuticals or just, human health or health in general. So.
Yeah, so very much on trend and a growing area.
Yeah.
And then what do you see on the tools side, or what are the new tools or the refinements to the current tools? Because in terms of data mining, data laking, AI, how is that playing a role in sort of the next generation of metabolomics?
Lots of different pieces there. AI, something we've, known about for a number of years. I sort of alluded to that the complexity of these pathway analyses are so complex. You just look at this one paper that, Jason wrote is you've got a pathway here, you got a pathway here, they cross over, what changes here is going to impact over there. And the more data we're able to generate, the more complex it is to get these interpretations. So I think something like AI is going to have a huge impact just in this space in interpreting and coming to conclusions or coming with ideas of what the next thing should be. Yes. On technology space in general, there's a couple things for us. We're always trying to just grow the number of compounds we're looking at. Some of these assays, we start with 20, they're at 100, now they're 400, 500 different compounds. So the more data that we can generate and the more sensitive the data, the more accurate the data, the better. So there's always new improvements in instrumentation, et cetera. And then just the work of our metabolomics team here in growing those assays. And then, just a little bit of a plug for us, the really exciting area that we're getting into right now is something called mass spec imaging. which is basically the same thing what we've done for Jason is, you're looking at metabolites, but now what we're doing is looking at that as a spatial context. So, similar to what a physician would do in a hospital, looking at histology, but looking at not just those cell types, but looking at how those metabolites are distributed, what's happening in certain areas of a tissue. So when you return to the gut question that Jason's been asking, and that those questions of the leaky gut and in between cells, is we could actually start looking at those tissues and say what's happening on a molecular level in the gut, in the cells, in the tissues, what's happening. So that's something that we've been growing in the last year or so. Something we've worked on for a number of years, but we've got some new tools here at the center that just have really game-changing in sensitivity and the number of targets we can start looking at. So,
no, really exciting stuff and cutting edge science, no question, in the human health arena, and I assume some animal health application as well, of course. So both areas very, very hot at the moment, that's for sure. And then in terms of the study and the publication, Josh, what do you feel some of the barriers were to overcome with this study? And because there's so much information to grab onto and grasp and interpret, how to make sense of all the data that's provided back, how was that managed and processed to really focus in on what a clinical study report for a study of this magnitude looks like, and how it's managed to a successful conclusion to provide back to the sponsor, and what has that done for so far two publications off this study? So Josh, do you want to speak to that a bit?
Sure. I think there's a number of challenges, and I can go in a lot of different directions with this. I'll do a few of those. Of course, as we already discussed, one of the challenges is setting the population at the front end, but part of that population then becomes also explaining in the consent form to participants what we're going to be doing here. When you're biobanking samples, that's a separate piece of the consent to be able to hold those samples, and what are you planning to do with them into the future? Because again, it's informed consent. So from the front end, we had to ensure that we were aware of what we were going to be doing with these, what potentially we could be looking at. I know this study didn't get into the genetics and things, but if we're doing genetics, then that's yet another consent form that gets added on. So, and there's always options for participants to take part in it or not. The other key thing here too is at the outset, we knew what our primary secondaries were going to be. It wasn't necessarily the metabolomics. So we focused the report around those other endpoints. So what was happening with the gut microbiome, how the stool consistency, again, all around the prebiotic claims. So the discussion on the metabolomics came downstream from the report. And that's where we got into the publication and taking this one step further. We had those biobank samples, running those outside of the report, but then turning that into publications. One thing just to also touch on too, which is quite interesting, is clinical research, as everybody here on this team knows, is quite expensive. So every time you add another biomarker, every time you look at another visit, another endpoint, there's money associated with that. And I think one of the challenges for the industry is not only knowing what the product's going to do, but how it does that. And that in itself can be quite costly to do, whether that's in preclinical models prior to coming into clinic or in clinical research, adding on these other biomarkers. The fact that we can look at these metabolomics more en masse, even after the fact, has a massive cost savings. If we were to look at each of these individual proteins or biomarkers in the blood, those could range from $1.50 a test to $300 a test. So that scalability is quite impressive now that the tools have come down in cost. The ability to get this information, it's a lot. It takes a lot to interpret. There's a lot of factors that you also have to consider. But the accessibility now to do more with less is a great benefit to, I think, all industries, as you mentioned, Dave, even to pharmaceuticals. We're all looking at this now because we're able to and get more out of this. From another aspect of this too, collecting adverse events, medical history is always important in a clinical trial. Ensuring you've got those documents in a case like this is very important. And Jason and I have had some discussions about some of the results and having to dig back into this because we know other environmental factors can influence these biomarkers. So looking at, did somebody have a cold or flu during this period of time? Was there seasonality issues that came into play here? Those are things that then we backed up, dug into to look at to see if we could explain some of these anomalies in the results. So very important to ensure the documentation is there throughout the clinical trial because you never know what questions are going to come up at the back end. So need to have the ability to go back, dig into these things.
Great. Thanks, Josh, for the insights. And as we wrap up the panel here, just one final question for each of the three of you, and thank you all for your input today in these various exciting but related areas. In terms of the next steps or what the future involves, we've touched on a little bit, but it'd be great if we'll just go around the panel and confirm what are some of the new key areas that, from your perspective and your vantage point to the world of nutritional science, what are you seeing next from your perspective? And maybe David, we'll start with you.
Yeah, so, I sort of mentioned that, you were asking questions on, nutraceutical companies and getting into this space, and it is still relatively rare. So it was really exciting seeing a company like Solnul get into this space. And I don't see any reason why all companies shouldn't be doing the same level of research and really understanding not just is there an impact of this, but how is it working? And I think it really helps companies come up with the claims and support it, and both regulators and patients and customers supporting that what they're doing is actually backed by science. So I think this has been an amazing adventure with Jason over the last 5 years. Looking back on this, I'd love to see additional studies. We obviously got something going on here in the gut, looking at the bacterial microbiome, the metabolites that are related to that. How is products like this impacting what the gut is doing? We know that bacteria in the gut have a huge impact on biology. Obviously, Josh, your question, it's cost and sample and time, but we are now able to start looking, like you say, at hundreds to thousands of compounds at relatively inexpensive, in the grand scheme of things, costs. So,
yeah, a ton of efficiency to pick up for sure. And then, Josh, what are you seeing from your perspective into the future on the nutraceutical research side?
I do see that this is an area that's going to continue to grow. Again, accessibility to this level of research is a game changer for the industry. Previously, research has always been focused on that claim. What does my product do? Achieving that claim. How it does it is often the afterthought or too expensive to get into. The other benefit, as we've also discussed, is what else could my product be doing? It's opening up that pathway to what else is my product doing? What other research could I be looking at? What other claims could I be moving forward to substantiate? And I think that's a huge opportunity with using technology like this.
Great. And then Jason, from your and Solnul's perspective, what do the next steps look like? What does the future hold?
Yeah, well, I think Dave and Josh have touched on a couple real key points. And one is that microbiome sample, right? So I don't know if I emphasized it, but we took blood and stool samples for Metabolomics. And we prioritized blood as the first study to look at how mechanistically the microbial metabolites are influencing the host and how is the host responding to that. So that was the initiative. But we still got those biobanked, those stool samples biobanked. And I think it would be really interesting to go back, because we've got so many changes in the microbes too, right? So how are the microbes communicating with each other? And to really look at whether metabolomics can give us a better understanding. We see bifidobacterium increase in response to Solnul, but we also see increases in Akkermansia. But Akkermansia feeds on host mucins. It doesn't feed on resistant starch. So there's an ecological interaction there that I think the stool metabolomics would actually give us a lot of insight too. So that's kind of one area that I think I'd love to pursue further. And the other, I think, that Josh touched on is, what else is my product doing? One of the real advantages that we got from doing metabolomics research is that it helped us identify future areas of clinical study. So just for example, the structure function claims we get from these biochemical changes are things like, may reduce collagen breakdown products. But from my perspective, it would be really great to bookend that type of claim with reduces fine lines or wrinkles, right? Some sort of patient-derived observation that really helps us position Solnul for example, the beauty from within industry. And the cool thing about the metabolomics data is that we had no idea that Solnul would do this. It was only by doing this research and asking that question, what else might it be doing, that it put us onto these really hot areas of future investigation.
Super. No, understood. Sounds like some other potential publications could be in the works downstream. That's exciting. It was great to have all three of you today and have you as well, Dr. Schibli, as a co-author on the publication, but also as provider of all of this insightful data through your institute. I really appreciate it very much. Josh, as always, great to talk to you about clinical trials in general. And Jason, great to have you on discussing what this means for Solnul as a sponsor and as an ingredient provider to the industry, and how cutting edge research and new age research can really assist in opening up new doors and corridors for you, and facilitating future research in some really vital areas of emerging and fascinating science. So appreciate your time today.
All right. Thanks, Will.
Thanks.